Welcome, my friends, to another episode of the Phenomics Podcast. I'm Dr. Cody Hahn, joined by Dr. Daniel Lawson and my good friend and colleague and fellow of the Molecular and Applied Science Laboratory at Auburn University, where we had the honor and privilege to do our PhDs together with Dr. Mike Roberts at Auburn. And so Matt Romero is a dear friend, is a very smart human being that I'm excited to talk with today. He's going to teach us a lot of different things in the area of genetics, and particularly the structure of DNA and how, well, I don't want to give it all away. Okay, so we're going to talk about DNA and the regulation of gene expression and the structure of DNA, and tie that into some exercise and perhaps some nutrition topics, hopefully with some practical applications. But really, this will be a somewhat nerdy conversation. So if you're not here for that, you might not want to listen to this because it's some pretty heavy molecular physiology, molecular biology conversation. But I think it's really fascinating stuff with a lot of exciting implications. And we're really honored to be able to talk with Dr. Romero and take some of his valuable time here today. So Dr. Matthew Romero, you are at UC Santa Cruz now. You did a postdoctoral research fellowship. Is that the correct title of that time at UCLA?
I think, that would be correct. Yeah, it's a long way of saying a 5-year research journey.
5 years. After that, 4 years of PhD, right?
And 2 years of a master's.
Oh, geez. So 11 years in graduate and postgraduate work. Impressive.
Yeah. And that was like 5 years is, just to give some backstory. In biology, the average postdoc, I think now, is either 5.5 or 6 years. So I actually finished a little sooner than what most people do. And there's a number of people who postdoc for 8, 9, 10 years sometimes.
Yeah, Matt, why does it take so long? Like, why is that typical for that area of research with postdocs?
Yeah, so I think it comes down to a couple different things. Number one is a lot of people are trying to understand, some very basic mechanisms of biological systems, right? And those things just take time to develop, like to develop your questions, to hone in your techniques. And there's also a lot of collaboration that goes on, right? So there's a ton of time that goes into that. I think the second thing that really is the biggest thing is you can repeat your experiments, right? So if something doesn't go right, a lot of the times you can say, well, I messed this up. I can fix it, or I can optimize it a little bit. I'm going to go back and do it again. When you think of physiology, especially the kind of research that Cody and I did, you had one shot, right? Like you had to have your ducks in a row. You collected your data, and then the data were the data. You couldn't go back and recruit another 40 or 50 people, or however many Cody would recruit for his studies. And say, okay, we're going to repeat all this again, except this time we're going to do 8 sets instead of 6 sets. You know what I mean? Like it's not very feasible. And so you get one shot, and so that means you're done a little bit faster. So I think those are some of the biggest things. And then the third thing, which is playing a role, but with the upregulation of biotech and how much they're recruiting PhDs and postdocs, is you're trying to get the best quality paper to make you more attractive on the job market. Your research paper, that's your productivity, that's to show how productive you are at your job. And so not only the number of publications, but quality. And so we pay a lot of attention to what's called the impact factor, which Cody, knows very well. And we've had tons of arguments on this back in the day. They're trying to get into journals that have higher and higher impact factors. And a lot of the times reviewers, when you send in those papers, will say, okay, this is nice, but we want X, Y, and Z. that takes another year. So it just keeps adding and adding and adding on.
Mhm.
And so that's what delays people from finishing. And it also delays people from starting their life. So there's a whole other, social aspect to this of getting, a postdoc and whatnot, because you're really asking these people to put their life on pause. And then if you have a partner, you're married or whatever, like you're asking them to also pause their life. So it's a huge time investment. And it's a lot of work.
Yeah. But it was worth it, right?
Yeah, I love what I do. I enjoy it very much. I like, because we would have these 3-hour conversations. We start at the gym, right? We'd be yelling at each other for an hour about evolution, let's say. We'd go to the gas station to get a snack after our squat session. We continue the argument there, and then we'd be in our offices, recruiting other grad students about this argument. And so I love doing that. And so as you progress, you start to do it in a more professional manner, but you still do have these debates and people are, critiquing your work and whatnot. But I love the challenge of trying to figure something out, learning something new. And, it's fun. It is hard, but but it is worth it.
Right on. Well, and buddy, you went deep into the matrix after our time at Auburn, and you have uncovered some incredible insights. And I'm excited to learn from you today. And man, I really think a good place to start that you have helped me to understand. How, I remember Dr. Roberts taught in the special topics class, the exercise genetics course. We talked some about the structure of DNA, but it seems to me that, from reviewing your recent work, that there's some more insights to be shared beyond what I had, previously known. Some of this is newer insights that I think it would be really cool for you to educate us on just the structure of DNA. And last conversation we had, Dr. Dan and I looked at its 2007 Bauman paper, where the K-means cluster and it goes differentiating individuals into response categories. And they looked, Matt, at in that one, they looked at myogenic gene expression. They also, we looked at Dan, mechano growth factor, IGF. MGF was one of the transcript abundance thereof, as measured by PCR. I have to be careful with my terms that seemed to differentiate responsiveness, where the extreme responders seem to display higher gene expression of mechano growth factor, mRNA. And one curious thing about that really is, well, why? And perhaps it's related to some of your research, right? With the structure of DNA and the various transcription factors that enhance, right, the expression of genes. So, buddy, I think that's a great place to start, is like an update on the structure of DNA and perhaps alluding to some of your research here on line 1. So long interspersed non-coding elements, and then these nuclear elements. Yeah. So take it from here, buddy.
Yeah. So quickly, I'll give a little bit of, background of how I got into this work, because I think it's important to highlight, how you can connect different fields of study, right? And so when I first started, I was doing very applied research. My my master's project was looking at like hand grip orientations with tricep activation. So, really kind of like how do we maximize muscle activation during different lifts. I did some very preliminary like saliva type studies, right, in my master's, and I didn't know what the heck I was doing in the lab, but was trying to figure it out. And then we both ended up in Dr. Roberts' lab, where we were like, oh, there's this whole molecular side. And so that was kind of my first insight into that different biological mechanisms are controlling exercise responses. And that always kind of fascinated me. I was always a little bit salty because I could never really get that jacked. And I was like, well, why the heck not? Like what is it that I'm not doing? And so that then whenever I figured out that, there's a biology underneath all this, it really kind of caught my attention. So it all started with that. And then with an observation that Dr. Roberts had made this one paper looking at line 1, right? So that kind of set off into gene regulation in general. And so maybe we can get back to line 1, at a different time, because I do really enjoy talking about the structure of DNA. I think we don't, in physiology especially, we have more of an understanding or an appreciation of genes now and pathways and whatnot, but we don't always think about exactly how the structure of DNA of chromatin is affecting our responses to things. And it's becoming more common. Like more people are doing this sort of research, but it's not as prevalent as, it's not the first thing that comes to our mind a lot of the times, like in a lot of other biological domains. So if you want, I'll kind of bring this first slide up, and we can kind of hit this basic thought process, and then from there we can build out, I think. So let me develop the simplest version of this.
We've had a couple.
So let me
recently on gene expression and differentiating, the sequence itself. So the possession of a gene from the expression of a gene, and eventually protein expression.
Yeah.
So DNA to RNA to protein.
Mm-hmm.
But really having the gene is one thing to understand, and the specific sequence of that gene. Then why do we see, for example, that in the paper where some people grew a considerably more muscle in response to the same trialing intervention? Why did they actually display more of these mRNA transcripts? Right, and perhaps it is related to what you to show us here. So this looks good. Enhancers at a glance.
Yeah. Enhancers at a glance. Yeah. So, when we think about our genes and how those mean, a lot of the times we're only thinking about the coding sequence itself, right? So those are your A's, C's, T's, and G's, your nucleotides, right? And, a lot of people think, oh, you have this gene, and a lot of those genes that they're thinking of may be associated with a certain disease. And so, you have this one gene. If it has a mutation, you're more likely to develop this specific disease. A lot of people think of it in those terms. And, exercise, for instance, there's been a lot of research in the last 20, 30 years about what genes are associated with exercise adaptation, with exercise responses, right? But I think it's important to note that this region, these protein coding genes is what they're called. They actually make proteins, which actually do the work in our cells. They only make up about 2% of our entire genome, right? So our entire genome is 3 billion bases. This only makes up 2%.
Let's just pause there.
Of those 3 billion bases.
Right, because I think that's one of the most misunderstood things. When I first learned that, my mind was kind of blown. Right, so 6 billion nucleotides approximately. And you just said that only 2% actually are protein encoding.
Yep.
So a very small percent.
Yeah, right. And this is, it is like almost jarring to hear. And I think the other thing is, right, like how much of those protein coding sequences we share with the most simple basic organisms, right? Like a lot of these across kingdoms and across, all these different animals and bacteria, eukaryotes, like we have a lot of the same genes, not every single one, but a lot of them. So it's like what actually separates us out from, these other animals, what separates us from, chimps or the rhesus macaques when they have a lot of the same genes. And a lot of them are so well conserved, it's really hard to tell the difference. And so that 2% is not as illuminating as we first thought when the sequences of the genome were being kind of uncovered or discovered. So, I think that's the first thing to understand is that these genes, of course, are important. They play a role, obviously. Like that's not to downplay their importance, but the rest of our genome, that 98%, has a big role as well. And the way that I study it is in how those non-coding regions, is what it's called, how they help regulate or control the expression of our protein coding genes. So, in the simplest form, right, we have our gene of interest here. We can say this any gene. It has this upstream sequence that's called a promoter. Okay. And so what that promoter does is it helps promote the expression of that gene. And it does so by binding a lot of what are called transcription factors. And so these are proteins that are floating around in your cell nucleus. So those protein coding genes, these transcription factors, will bind to the promoter, and they recruit a ton of other proteins to bind there as well. And we have what are called polymerases, which bind here and then transcribe or copy this gene from DNA into RNA. Okay, and that kind of starts the process of gene expression. So you and I think you and Van last time I watched that one talk about a little bit of the central dogma, right? Like so DNA to RNA to protein. This is at the level of DNA to RNA. That's mainly what I focus. Now, these transcription factors, these are sequence specific, right? So what that means is they recognize very specific nucleotide sequences. And so these promoters have kind of evolved to have specific sequences that help promote these transcription factors. Now, that's in the most simplest sense, right? So here's your gene promoter, there's your gene. They're very close in proximity. We're saying anywhere between a few hundred base pairs to maybe a few thousand base pairs. That's all in the vicinity. You get expression of your gene. That RNA then gets made into a protein, and then that protein goes off and does some function. What was discovered in the 80s is that there are sequences that are away from this gene, either downstream in this case or upstream from that gene, that also can affect how much RNA you're getting. Okay, so now you're not just talking about a gene being expressed, so being on or off. You're talking about the actual quantity of a gene being made. And that's where these enhancers can really come into context. So these enhancers, like I said, be very, very close. They can also be very far from your gene of interest. And so we're talking, few thousand base pairs. It could be tens of thousands. In some cases, it could be like 500,000 or a million bases away. So it can be a pretty large distance away from this target gene. So, when these were first kind of discovered, you're like, well, how the heck can this sequence that's way over here, how could that actually affect this gene that's, a million bases away from it? And kind of what's come to light these last, 10, 20 years has been that these enhancer sequences will actually fold in 3 dimensions to their target gene. So what does this mean? Like, what is this enhancer actually doing? This enhancer a lot of the times is doing what a promoter is doing, right? In the most general sense. So it's recruiting transcription factors, it's recruiting polymerases, it's recruiting other, histone modifying complexes, which we'll talk about here in a minute, that help aid that communication between the enhancer and the target gene of interest. So when you're doing that, you're essentially like increasing the concentration of your transcription factors in your polymerase in this three-dimensional space, right? So by increasing that concentration, you're increasing the probability that you're going to be expressing a gene. And because these, all these little molecules, they're in this little vicinity, they're more likely to interact and they're less likely to kind of go away from there, right? From your area of interest. And so you're constantly having like this gene expression kind of cycle taking place. And then eventually that kind of dissolves, which is kind of an open question right now in the field too. It kind of dissolves it. And now you obviously decrease gene expression because you've decreased the interactions that are happening between this enhancer and your target gene. So I'll pause there, Codes, because I know you love questions. And so I want to make sure that we're on the same page right now.
That was a beautiful explanation. I think, man, one thing I'm reminded of some of Adam Sharple's work on methylation and how that seems to be, in some sense, preserved. You have an 8-week training intervention, and then you detrain for at least as much time, if not more, and you return to that same style of training. There seems to be this epigenetic memory. And the transcription factors, Matt, I think it would be cool to give an example, especially in my meathead brain, to connect some dots here on perhaps like the mTOR pathway. Could you help us understand, like, or another good one might be like ribosome biogenesis, just as based on muscle growth. Is there an example here, like of a transcription factor or transcription factors that seem to be upregulated in response to resistance training that could influence protein expression that relates to this process, just to connect some dots?
Yeah, for sure. So when you're talking about resistance training, in the context of let's stay with the context of ribosome biogenesis, you're mainly thinking about a transcription factor called c-Myc. So c-Myc is also kind of an end product or a transcription factor responding to the mTOR pathway as well. So c-Myc and mTOR and that signaling has been investigated quite a bit, as we both know, in cancer research, right? Like it's a growth pathway. Cancer research was a huge area, just general evolution of how these pathways evolved and came to be has been a large area of research. So that's actually a very well-studied pathway that more recently has been focused towards resistance training and skeletal muscle hypertrophy. But that's one transcription factor where you have, this mTOR signaling, you have phosphorylation, all these, kind of intermediate steps, and then you end on your c-Myc protein. That transcription factor then will bind, I'm showing this example, this one gene, but c-Myc will bind all over the genome. And this is true for all transcription factors. This is a probability-based mechanism. This is not so much a, 1-for-1 or a binary mechanism. Like it is binding all over the place. And a lot of the times it's binding where you have sequences of DNA that are exposed to the environment, let's say. So kind of trying to tie in your Sharple's point with c-Myc and transcription factor binding. You mentioned DNA methylation. DNA methylation largely works at a gene promoter, and it's associated with gene repression. So a lowering of gene expression or turning off of how much a gene is expressed. And so what that does is that DNA methylation will bind. It recruits, the DNA methylation will take place where you actually will add a methyl group to a DNA base. What that does is that essentially opens you up now to binding with a ton of other proteins that recognize that methylated DNA, right? And so that's a huge factor in this, is when you make a change like that, you're not just changing the DNA itself, you're also changing what's going to interact with it. And so it's a multi-layered approach. So what ends up happening is these proteins will now recognize that methylated DNA, and then it'll work to recruit other proteins that come in to really bind up that region and close it off, right? And so in doing that, that's how it turns off this gene, is it becomes less accessible to these transcription factors and polymerases. So in this example, it'd be less accessible to CNIC, right? And the same thing can happen at enhancers as well. But largely you're looking at, instead of DNA methylation, you're looking at histone modifications. So what are histones, right? So histones are proteins that DNA wraps around, and it helps stabilize DNA. And then it helps it, in this case, more, reactive to the environment. Okay. So we can think about it that way. So when this DNA and histones are bound, now these are called nucleosomes. Nucleosomes are the collection of a histone and the DNA that's wrapped around it. When you modify these histones, you can have different effects, right? So it's not like DNA methylation. DNA methylation, for the most part, in general, is gene expression is off. Histone modifications are much more nuanced and context dependent. There's a few review papers called, that are talking about histone codes and what it means to have different histone modifications, and I've highlighted two of them here. So one of them is histone 3, lysine 4, methyl 1. So what is that? A histone is not just one protein; it's a collection of proteins, and they bind. They have they're named H1, H2, H3, H4, right? And so you have this specific histone, and you have this very specific amino acid residue that's being methylated. Okay, so you're actually methylating a protein on here. Now, in this context, it's the opposite of DNA methylation. When you add H3K4 methyl 1, you're actually relaxing chromatin. Okay, so this is why I was saying the nuance is important here. And on the other side of that, you have histone acetylation. And so it's still on H3, but now instead of lysine 4, you're on lysine 27. And so this also has a more relaxing effect. So when you see this, the nucleosomes are kind of creating more space, more DNA is being exposed between histones, and now this provides an opportunity for a transcription factor to bind and to recruit those other factors like polymerases that I was talking about. And so that's how this actually has an effect. So you kind of need both things to happen. You need this relaxing chromatin. You need a transcription factor to come in there and bind in order to maximally express this gene. How are we doing now?
Yeah. Well, perhaps this is jumping ahead, but augmenting methylation and acetylation in a targeted way through exercise and/or pharmacology and nutrition or supplementation. That's 4 or 5 different things, but perhaps can you give an example of how we could augment methylation and acetylation strategically to promote muscle growth, for example?
Yeah, so I'll go back to your Sharples example because he had a really good study design. But this will be DNA methylation, and then we'll get back to histones here in just a sec. So Sharples had this really great study design where he had people come in, they resistance trained, right? And then they stopped resistance training for a number of weeks as like a washout, right? Then they had those same individuals come back and resistance train again. They took muscle biopsy and they looked at DNA methylation at each one of those time points, right? So what they showed, so I mentioned that DNA methylation here can repress gene expression. Well, they found that genes that are important for muscle growth, there was less DNA methylation, right? So that means they're going to express genes more, and they were in this case. What he found is that the people that had resistance trained and then stopped and came back, they maintained that lower methylation status. And when they resistance trained again, they were able to more robustly increase the expression of their genes. Okay, so this is getting to what a lot of us heard in sports, right? Is that your muscle had a memory. And if you learned how to do something, your muscles would remember what it was doing, and you'd basically still be better than you were when you first started. And so this is kind of happening in this context, but with muscle growth, is that the genes kind of remembered that you had done activity. And so they had their genes ready to go. So when you brought in those signals, in resistance training in this case, you're able to express those genes, right? And so that is kind of at the basis of all of all these adaptations is, to a sense, gene expression. We can talk about the nuance of, protein modifications and whatnot too, but, in terms of how just in general animals adapt, whether it's to, the environment, to growth, like these kind of things, like gene expression is a huge player in this. So it's like it's remembering it. And so when you think about histones now, man, it's really awesome that we don't know a ton about what happens to enhancers and this non-coding region of DNA, right? So there have been a number of articles now, a number of groups that have looked at what's called accessible chromatin. So that is kind of agnostically looking at what regions of the genome are open, okay? From there, you can kind of filter down. You have your open regions of the genome. You can then say, okay, well, which regions of these are protein encoding? So you can kind of filter those out. And you can say, okay, these are open, so these are more likely to be on. These are closed, these are more likely to be off. By filtering those out, now you look at everything else. You say, okay, all these non-coding regions, which ones are open? Okay, these are open, they're more likely to be enhancers. But that's kind of where our interpretation is limited, because just because you have these regions open and they may be enhancers, you don't know what they're targeting. They could be targeting this gene. They could be targeting one that's way down there. They might skip 2 or 3 or 4 genes. So you have to really tie them in 3 dimensions in order to get down to what's actually controlling your gene of interest. And that's where some of my work is what it's trying to do. But that's right now the limit of where we're at with exercise. And so that's, I think, the exciting part is there's still so much to be discovered here. We know a lot of the transcription factors that are involved with exercise response, right? We don't always know where they're binding because these transcription factors are cell specific. Okay, now what that means is your muscle tissue is going to have a suite of transcription factors that your liver is not going to express. And so those are going to bind to very distinct places. Not only that, let's say you have the same transcription factors that are expressed. They may bind different places to regulate different genes. And so, like I said, it's probabilistic in this model. So you really do have to get down and say, I have to look at this specific tissue type and under this specific condition in order to understand properly how these genes are being expressed and what's controlling them upstream. So upstream meaning their transcription factors, their signaling pathways, their, plasma membrane receptors, all this sort of stuff.
Well, yeah, that's fascinating stuff. It reminds me, Dan, of the paper that you reviewed recently, looking at period of training followed by detraining and then a return to training, and how the continuous training group versus the periodic training group, right, Dan? The same point, but the slope of the line in terms of muscle growth appeared to be steeper in the group that had the cessation of training and then return to training.
That's right.
Right. When they did that, and perhaps that's what's going on here. But Dan, that paper, was it like 8 weeks, 8 weeks, and 8 weeks? Do you recall?
It was the continuous resistance training protocol was 20 weeks, and that was continuous. And then the group that took the training break, it was a 10-week training period followed by 10 weeks of non-training or detraining, followed by another 10-week training period. And so, yeah, especially the retraining phase for the one group, the first 5 weeks of that 10-week retraining period, that's where there was that really steep slope with that return of muscle size and strength that was much faster than the group that had done the continuous training. And I was thinking about that paper too, so I'm glad you brought it up, Cody. Was exactly where my brain was going with this. And then that makes me wonder, like, how long does the muscle memory effect last? Because I think that was one of the longest training breaks. It's not like a disuse study where they were working out and then they just didn't work out. Like 10 weeks is pretty long. I don't know if there's studies that have gone longer than that. I'm not sure. But like, how long would that last before that it's the suppression, right, of methylation on the promoter side of the gene, if I'm understanding that correctly? So I guess
In this case, definitely.
Yeah, that's exactly what I was wondering, Matt. If you had some insight on it, perhaps half-life isn't the right word, but like how long is that demethylation state maintained after a period of training?
So it can be quite a while. So I can't really see your guys' faces. So just for a second, I'm going to stop sharing. We can go back to it. DNA methylation can last a long time. Okay. So in terms of exercise specifically, I don't have a great answer for how long it's seen to last in humans, but let's think about this, kind of, outside the box, or think about other biological mechanisms. So bionuclei are terminally differentiated, right? So we know that, which it means these nuclei are not dividing. They're not making more. Okay. So why that's important is whenever you divide, during that process, when you split your copies of DNA, you have to make sure this side looks like that side, right? And it doesn't always happen. You don't always have the two sides looking the same. And so that decreases your chances of that DNA methylation remaining there, because it may not be posited again on this side, right? So we don't have to worry about that, right? So that means we're going to keep these signals a little bit longer, I would think. The other thing is DNA methylation is what's called an active process. So you add it, another enzyme has to come in to remove that DNA methyl mark. So why that's important is because that takes a certain amount of expression of that gene. That's going to take a certain amount of substrates that are available. But not only that, when you remove that methyl group, it doesn't make the base is not the same. It's an enzymatic process. So you have, in this case, you methylate cytosine, right? You don't just remove it and now you have an unmethylated cytosine. You have this like intermediate base that you need to further modify in order to get back to that cytosine. And that's done by a separate enzyme. Okay. So it's now this sequential process. And so it's an active process that needs to take place over time. So those methyl marks can last there a while. Now, in this case, we're talking about the opposite of keeping those genes from being methylated to ensure kind of growth gene expression at a later time, right? But the same kind of idea is there, is that you still have to have these methyl depositing enzymes available. They still have to have a signal to recognize those promoters in order to methylate them, right? And so that all takes time, and that takes signals from the environment. So if you're not getting these external signals like that, that state could last a while because you're not differentiating, because you're not having these external signals that say we need to methylate this gene. 10 weeks, maybe it's 6 months. We've talked about training before, and this is not talking about DNA, but we've talked about satellite cells, right? And you can add satellite cells to your muscle after training, and you can remind me like how long that stays around. I think it stays around for a long time, right? So
it needs to be retained for quite some time. Years.
Yeah. So, right. And I know there was some early studies. I haven't revisited the literature in a while, but like people who used anabolics, right? They add satellite cells, and those seem to stick around for years after they finish taking them. So I would imagine that these signals can stick around for quite a while. But I would think it'd be a function of how long you've trained, how hard you trained. I think if you train for a week, you may have some DNA demethylation, but I don't know if that week is long enough to kind of like train your system. And that's kind of how I think about this stuff, which might be naive. I'm not like, this is a whole new area of research that we're kind of undertaking, but I think that, there's kind of these gene networks associated with environmental responses, in this case, exercise. And if you don't continuously stimulate those systems, they may not last, right? And so if they're not lasting, then you may remethylate those regions. But I would think you'd have to do it multiple times. And so maybe 10 weeks of training is a good stimulus, and then you get 10 weeks where it stays there. And maybe it's a 1-for-1, right? If you train 20 weeks, it would take 20 weeks to revert those changes back. And so that would be an interesting study. And admittedly, that's something that's kind of missing from the field are these detraining and then retraining studies, because we know exercise is good for you. We don't always know why mechanistically in terms of at the DNA or the chromatin level, but we obviously know it's good for you, but we don't know how long those stay around. And if you were to retrain, how long does it take you to kind of get those benefits back if they went away?
Yeah.
Yeah, I guess one question I would have with that is how important is the count of mononuclei for that retraining phase? So if you're to detrain and then go back to a retraining phase, having more mononuclei in theory makes sense that it would be important, but is it?
That's a great question. So I guess how good versus necessary, versus optimal, like those are all sort of like the nuanced part of the question. So we know, right, from John McCarthy and Charlotte Peterson's work out of Kentucky that you can grow without satellite cells, right? Now their model was a overgrowth model where they ablate some of the muscles in the calf and then those muscles continuously grow. But that's not necessarily an exercise signal. But nevertheless, there's other studies kind of supporting this idea. But where satellite cells are definitely needed is during regeneration, right? If you don't have satellite cells, you damage the muscle bad enough, the muscles don't regenerate well. So I bring that point up because those satellite cells are donating nuclei, right? And so that you're increasing the number of nuclei or replacing the nuclei that were damaged during, in this case, exercise. So why I would think that you would need them in order to upregulate that transcriptional response, right? So the proteins that you make are kind of based on how much RNA you're expressing, right? Not all the time; those numbers aren't perfectly correlated. The more you express a gene, in general, the more likely you are to see that protein product or more of it. So I think if you have more nuclei, you're expressing more of that gene, presumably, you're more likely to see more of that protein. And so I think that would make a difference. But from that same group's work, when you knock out satellite cells, the mononuclei transcriptionally respond and they increase their expression over what they would normally. So there seems to be a built-in sort of compensatory mechanism. So, again, optimal versus needed, those are two separate questions. But I think it would help for sure to have more nuclei. That's my opinion.
Yeah, that makes sense. Did you have a follow-up, Dan?
Actually, my brain kind of went to, I thought about when we were talking about the Bauman paper, and we made the distinction that gene expression, that an increase in mRNA, if we see that, doesn't necessarily mean that there's going to be an increase in the amount of protein.
No.
I guess why wouldn't mRNA be translated into protein, even if there's a pretty significant upregulation?
So this is the fun part of gene expression. It's frustrating, but this is the fun part. There's so many mechanisms that are at play to keep kind of homeostasis, right? So translating mRNA into proteins is obviously an energetically costly event. And so the cell just doesn't do it willy-nilly, right? But also the level of gene expression could dictate or change what type of cell you're looking at. So I'm particularly thinking of development. In one cell type, if you express more of this gene, you make an entirely different tissue. Whereas if you express less of it and then you make more of this separate gene, you turn into this tissue, right? So the amount you make matters in development. When you're thinking, in this case, mature muscle, that's not so much what we're worried about. It's more so like, what do we need to respond, I think? And so a lot of the times you have these various mechanisms like mRNA decay, where you actually have, these non-coding RNAs or different non-coding RNAs that will help degrade those mRNAs, right? And so that keeps it from being translated. So even though you have an expression, you may also have a decrease in the degradation of the mRNA that's out there. Now, another thing, this is what I think is my opinion. This is what I think is more so the case, and that's the timing. When you express a gene, it does not mean you're going to make that protein at the same time or even shortly after, right? So those time differences I think matter, because we can only collect our muscle samples so close or so far from the exercise bout, right? And so this is kind of what's been limited in the human studies that we have, is we have predetermined time points, 0, 2, 4, 6 hours, whatever. The expression of that gene may maximize at 4 hours, but its translation may not maximize until 12 hours. You don't always know what that line is, what that response is. And so I think that's at a lot of the differences that we see. And there was actually a recent paper that came out of Switzerland, I believe. It was a 2023 paper. They looked at, in this case, endurance training. They looked at mice. They showed that animals that were trained had a different gene expression response than animals who were exercise naive. And some genes turned on sooner, some genes turned on later, and their amplitude changed. So some expressed even more when before they were trained, and some expressed even less. So there's all of these sorts of subtle changes that we're not accounting for a lot of the times. And so I think that played a role in the actual amount of protein you're seeing at this specific time point. So I think sometimes it's a technical variation, although not always, right? Like you can see a gene be expressed but not be translated, and that might be due to those intermediate steps that I was mentioning about the degradation of the mRNA.
Matt, I'm also reminded of, try to tie some of this to just some practicals, that some of Dr. Karen Esser's work around how certain genes seem to display expression patterns based on a circadian rhythm. Regardless on the stimulus, they just have this characteristic expression pattern across a 24-hour period. And then I'm aware of, I want to say, Matt, you would know, is it Johnny Camara out of, it was in Australia, but I believe it done some on this and kind of alluded to resistance training during the perhaps the accessibility of transcription factors to the theoretically enhancer regions. You could augment the expression of certain genes to promote protein synthesis by training later in the day versus in the morning. And we tend to be stronger in the early afternoon or evening as compared to earlier in the day. I'm not saying that's all genetic, but do you have any insight on that? Like if some of these important regions for adaptation to resistance training are perhaps more accessible during certain times of the day and more receptive to the stimulus of training to augment expression.
So I'm familiar in general with what you're talking about. Like I've seen some of that data, but I don't, off the top of my head, I can't think of examples directly testing that. But in other systems, I do know that have looked at kind of diurnal rhythms and different tissue types that some regions become more or less accessible throughout the day, right? So it can happen. What'd be interesting is, I guess let me back up, right? When you're talking about accessible regions, these regions can be accessible, but they can also be made accessible by something. So there are these proteins called pioneering factors, right? So these are like kind of specialized transcription factors that go in and they help recruit histone modifying complexes that help open up chromatin to allow other transcription factors to bind, right? So it could be, I think something that's interesting is, maybe later in the day, let's say we've been fed, so we've eaten. So now you're having an upregulation of all these, food-related pathways in muscle. You perhaps can be turning on some of these pioneering factors that are making some of these regions more accessible, that allow these genes to be I guess, accessed, right? Now, so the main transcription factors in muscle that I'm familiar with are clock and BMAL, right? Those are kind of time of day responsive transcription factors. Now, whether those are pioneering factors themselves, I don't know. I don't know if those are actually able to come in, open up chromatin and allow transcription factors to bind, or if there's something upstream of them slightly that is opening it for them that then allow them to come in and express those genes or set off their own signature way or gene network. But it would make sense to me that, and almost think about it in my mind, again, this is just me thinking, like the chromatin itself is almost breathing throughout the day, right? So it's like for a short amount of time, you're well fed, you're well rested. It's like it's breathing out like, oh, I can relax. And then towards the end of the night, for whatever reason, whether it's food related or just, it is time of day related, that chromatin then closes back up in some regions that allow you to kind of progress in your physiological adaptation or stop signaling pathways from being overstimulated. That's kind of how I think about it. Whether that is how it's happening, I'm not entirely sure, but I think that it sounds like a fun medicine for me.
So we were talking about this kind of breathing effect and accessibility, and perhaps some of that's mediated by light exposure, nutrition, exercise. We can modify it through those stimuli. But man, I'd be curious to know if we're trying to, in your area of research, like and perhaps just future directions with that whole thing, do you see this being an opportunity to develop pharmacological interventions for targeted gene expression? Or do you see it more from an exercise and nutrition intervention standpoint? Like, which of those are you kind of thinking this area is going to take you? And I guess moreover, what direction do you think is most appropriate for us to go? Like, are we looking more, perhaps CRISPR technology isn't the path, but you mentioned like epigenetic modification through pharmacology. Is that kind of where you're thinking in this, or is it more exercise and nutrition related that we can figure out how we can use those behaviors to modify gene expression?
Yeah, so I think those two things kind of feed into each other, right? So on one hand, I was mentioning, how much of this can we actually control? What is under our own power to do? So if we exercise at this time with this amount of food, with this specific macronutrient content, does that kind of optimize what we have, right? So is that going to ensure, or at least, improve our chance of building more muscle or adapting better? And you'll always have a subset of the population that wants to do that, right? There's going to be people who want to push themselves, or that's just like their hobby or their therapy. And so I think these sorts of insights will always help them. But really, for most people, it's too much information, right? Like it's they want to be healthy. I believe in it, but they're like, okay, do I do Pilates or HIIT training or CrossFit or powerlifting? Like they get all this gunk, and we've talked about this before. Like that always drove me nuts. And that was, probably worse than ever with all the influencers. Like everybody's got an opinion. And these poor people just want to get healthier, right? Like, they don't want to be on the Olympia stage. They just want to be able to run around with their kids and go on vacations and not, be miserable when they have to walk uphill. And so, I think that is kind of at the heart of what we're trying to do is what's the best way we can help these individuals? And so I do think, naive or not, I do think there are special things about exercise you can't recapitulate with, and they've shown this with just weight loss, for instance, right? And there are benefits you get from exercise that happen independent of weight loss. So obviously strength, you don't need to lose weight to get stronger. You don't need to lose weight to increase your endurance. You don't need to lose weight to improve your metabolic profile, right? So your lipid content, cholesterol number, those kind of things. You don't need to lose weight for all those. It helps, but you don't have to, right? So there are special things about exercise that are unique to exercise. Right? And in that is some of those kind of cell-specific or tissue-specific things I was mentioning before about enhancers and where transcription factors are binding. So I'm going to, I'm going to share one more slide because I think it's pertinent here. So let me back here. Okay. Can you see that okay? So here is kind of a rundown of ways that enhancers can be either mutated or modified and what impact that has on gene expression. So this is going to get back to that point I'm talking about. So you can have your gene that is regulated by one enhancer. Okay. Now there are mutations that can happen here. So a mutation would be a nucleotide-based change. So maybe you have an A there, now it's a T. That affects how a transcription factor binds. And so in turn, that affects how much gene is expressed. Right. So you can have this sort of thing happen. You can duplicate your number of enhancers here. You can have a change to your overall 3D structure, which we haven't talked about, but could happen, that allows extra enhancers to have access to this. So again, it's all probabilistic in a sense. So a lot of these things can be changed, right? But on top of that, what is unique about enhancers is that they're also very cell type specific. So you might have an enhancer that is active in brain tissue, right? You also might have enhancers that are active in every other type of tissue. In this case, is the developing, mouse in this case. So you have the same gene, right? So let's say this is CMYC. We talked about CMYC earlier. In the brain, it's regulated by these enhancers. But in, let's say, the developing embryo and other parts, the limbus or the tail, you have different enhancers that are regulating that same gene. Okay. Why does this matter? Well, this matters, A, because it shows how biologically how specific everything is, how these things have evolved to use the same gene across different tissues for how those tissues need to work. But what it also allows is for a tissue-specific sort of modification or modulation of gene expression, right? So this is really kind of at the heart of a lot of the new, pharmacological techniques and gene editing techniques that are coming out now or being developed. Is sure you could use CRISPR to mutate these regions, right? Well, let's say you wanted to change the sequence of this enhancer from one thing to another to allow it to work better. But to do that, you're having to change the sequence itself, right? So that's probably going to happen in a ton of other tissues as well. So that's going to dysregulate all their expression. Now that may be helpful in cancer. Let's say there's a mutation there in cancer and you want to fix that with CRISPR. Got it. Like that makes total sense. But if you're talking about fine tuning gene expression, which we talked about was important for development, it's also important for evolution, right? Like, we think about, there was a study done with chimps, and the amount of, they express a lot of the same genes, but the amount of those genes that they expressed is higher in their muscles than in ours. And so they in turn are much stronger because they have more of that protein content as well. So fine tuning the expression is important. So what you can do instead of using CRISPR-Cas9 to mutate the regions, then we have what's called CRISPR-D-Cas9 or dead Cas9. So that means the enzyme that's responsible for these mutations is not active. It does not actually mutate the DNA, but it does bind to DNA. And what it does, instead of changing the sequence itself, it changes those histone modifications that we were talking about. So now that region becomes more accessible and it becomes more repressed, and now you can fine tune that gene expression. So if you can fine tune that gene expression, now you might be able to fine tune that response. And so this was actually done in obese animals. They targeted one gene. They looked at the gene promoter. They noticed that it was the promoter was more highly methylated at that region. In this case, it was histone methylation. I used CRISPR-Cas9 to demethylate that region, and the animals lost weight just by changing how much that gene was expressed or repressed, right? So that gives insight into how these things can be fine-tuned. Now, we're kind of decades away from that being a viable treatment or path to take, right? But these things are happening very quickly. There's other things you can do. You could actually target RNA as well as CRISPR Cas13. So that targets the RNA. and you can degrade it that way, or you can modify the RNA, and that can affect how well it's being translated. So that's a whole nother region of gene expression that we haven't even touched on. Now that specifically is outside my expertise, but that's a whole nother layer that's fun to talk about. But so, I think now on top of the actual sequence mutations or modifications that we want to make, there's more and more technology being made to modify the epigenome so we can modulate that gene expression. And I think in general, we can develop tools that do this for exercise, not for trying to get Mr. Olympia, right? Like, although that'd be cool, but to actually help people get these positive benefits from exercise that either can't, maybe they're bedridden, maybe like, dude, some people work 14 hours a day, right? Like you have to go home and see your family. Like I get that. So can we develop these tools that are accessible to these people that allow them to be healthier? Because recently, right, like we were the first generation that are living less long. We're living shorter lives than our parents, right? Like so, our health span is decreasing and our lifespans are decreasing. So, how can we optimize those things? And I think exercise is an excellent tool for that. For understanding how it happens, like how you actually maintain healthy cells, healthy tissues, healthy bodies, but also for the non-property sort of regulatory pathways that may not be active in other conditions, whether it be in a disease setting or just in vitro with cells, or under just heat stress. Like there's so many different things that exercise does because it does tie in all these different pathways and they converge a little bit differently than they would in these other more well-studied scenarios.
Well, that was really intriguing with what you just showed with the one gene, but different enhancers and different tissues. So were you saying that?
Yep.
This is a stupid question, but if this was a quiz question, I know how I would answer it. Are the sequence? Are you saying that the sequence of DNA is actually different in different tissues?
And so, okay, very good clarification. So the sequences themselves, right? So whenever we are born, let's take it back to the etiology, as you like to say. When we are born, our DNA, every cell has the same, more or less, there'll be some mutation, more or less has the same exact sequence in every cell type, right? So these enhancers are there in every cell type. They're just not being used. And that is because of the mechanisms that we were talking about earlier with the histone modifications, accessible DNA methylation, those sorts of things. And so chromatin is controlled in this very cell type specific way to keep enhancers either on or off in order to properly control the expression of the tissue specific genes, right? So the sequence themselves is not different here. That sequence, even though they have different colors and they're different number of it, the sequence along this linear track of DNA is the same in these two examples. Again, the only difference is that this one is active now in let's say the yellow one is active in the developing tail. That's not active in the brain. That's the only thing, is that it's accessible, doing its thing, or it's repressed and not doing much.
Well, and that's okay. So now, Matt, the sequencing.com, for example, is a company that you can sequence your entire genome after a buccal swab, the nuclei and buccal cells, just to get the source code. And then, there's different things you can look at, obviously having all of that information. And now there are tests that one can do to look at methylation patterns. But it seems to me, what you're saying here is that perhaps the most appropriate way to explore that is tissue specific. It's looking at, you'd have to do biopsies, right? To understand, what I mean? Like there's people that are talking now about, well, you can do this buccal swab or do a blood test. And if you have the MTHFR mutation, that you're not methylating effectively in the liver, therefore you should take these B vitamins, these methylated B vitamins. Can you actually confidently say that from saliva or blood, or would we really need to look in the liver?
I mean, I would think you'd have to look at the lip, honestly. I think these things, again, you're talking about probabilities. You're talking about enzyme kinetics, and I don't know. Maybe they know the maximum, efficiency you need from an enzyme in order to methylate properly. Maybe that small change in MTFR gene is not decreasing the enzyme kinetics so much that you can't methylate things appropriately, or maybe it does, right? Like so those are things that we don't necessarily know that we would need to see, right? And so these studies may have been done already in mice where you can make small amino acid changes that changes how efficient the enzyme may be, and then you can track, let's say, DNA methylation with this. And so that is one thing you could do. But if you really want to understand, you'd have to get to the tissue specific level. And I think that's incredibly difficult, obviously. How are you going to get a biopsy of the liver or the kidney of somebody? And so a lot of people are trying to get proxies of this, right? So you have people who are looking at the vesicles that maybe in the blood that carry a specific transcript from different tissues and give it insight of, how well is this tissue working? What's being expressed? Is this a proxy for something? And so that's kind of where a lot of people or a lot of labs are kind of going, is how can we look at it indirectly? But if you're, really thinking about from these tissue specific mechanisms, then you have to look at the tissue. The DNA sequence only takes you so far, and it tells you a lot of information, obviously, but you're talking about risk and you're talking about odds, right? At this point, and that only goes so far.
So you wouldn't feel confident saying that one could do it's a popular example, look at MTHFR and be like very confident that you could leverage that information to influence methylated B vitamin intake. We would really need to understand more about what's happening directly in the liver, and moreover, what's the outcome variable? Like maybe there's blood biomarkers that we could look at, but it's not as simple as just knowing the source code itself.
Well, I think it is context dependent. I think if let's say you did these studies in vitro or even in vivo with mice, right? And you showed, okay, we had this mutation, we gave B vitamins and they did end up methylating more, or they recovered their methylation status. I mean, if you're showing things like that, then it's, I think, a pretty safe assumption to think that it's a good practice to do. But it's still mice to humans, so it is a little bit of there is some chance that it doesn't work exactly the same. And then there's some contexts where, like, if there's a gene mutation, you're going to have this issue, right? Like there are those contexts as well. So I think it's useful information to have, but I don't know if it's quite as simple as just you have this SNP or this, variation. If we do this, it'll fix it. I think that's really hard to show evidence of. And not only that, but if, you take in the B vitamin, does it get to your liver? Like, does your liver show increased levels of, the vitamin and the cofactors and the intermediate steps that's there? Maybe it does. I don't know. I haven't looked at this so closely, but that's just another layer that you have to think about. Of is it actually getting to where you want it to go? And the liver sees almost everything, so probably does. But these are just some of the things to kind of verify, I think, before moving forward to these sorts of kind of over the counter, but like self motivated types of things.
Man, well, Okay. Kind of going back to those perhaps 2% of our DNA being protein encoding. Talked about in enhancer regions and transcription factors and how gene expression is regulated. Matt, you know, done a lot of work on line 1. I remember having conversations at Auburn about this. You've been thinking about this for a while and then published that excellent review. That was 2 years ago that it actually was in print, right? The 2023 paper. Yeah, and that was, you had put a lot of years into that, man. Just a powerhouse of an article. Line 1, man. Line 1.
Yeah.
So of that other 98% of DNA, how much of that is line 1? And I guess, can you just define line 1, give us some context? And then I did want to discuss line 1 as it relates to exercise, how exercise can affect that, and particularly how line 1 may be playing a role in pathogenesis.
Yeah, for sure. So let's, this is here's kind of an intro slide on that. Can you see that slide? Okay, so here's obviously a pie chart of all the things that are encoded into our genome, right? So here they have protein coding exons. So how you quantify, or what you call a gene versus protein coding genes, right? Those are a little bit different because you could have non-coding RNAs that still do something. So those can be categorized as genes. So the numbers that come back here that I was saying, that 2% can vary, and some people categorize it differently. So it can be 1 to 2%. So there's some variation there. But here they're having our protein coding exons as less than 1% of the entire genome. Then they're categorizing all the different regions or different types of DNA that we have. And as you can see here, almost half are what are called repetitive elements, and there's different types. And so you have lines, either long interspersed nuclear elements, what they're called. These are like ancient DNA that we have that, from simple eukaryotes, they have some of these. These have basically been found in almost every organism alive. Then you have signs, which are short interspersed nuclear elements. You're looking at about 11% of the genome. Then you have retroviruses, either kind of dead, and they are similar to retroviruses that have made their way into our genome. These are these are kind of like parasites. And then you have DNA transposons, which we can talk about the difference here as compared to lines. But these are a region of the genome that you can actually move around, and we can talk about that next. But then you have a protein coding exons, and then you have this other map. What does that do, right? So this is a lot of everything else. So you're talking about your regulation of your non-coding enhancers. You have regions that encode for different sorts of RNA. And so that's largely been called like the dark matter of the genome. And we're still trying to figure out what all of that does. But here's, when you talk about lines, right? Almost a quarter of our genome is made up of line elements. And so there's lines are a very specific class of what's called a transposable element, right? So a transposable element is something that can move around in your genome. And so that sounds kind of scary. Like, we actually have genes that can move from one place and then they go somewhere else in our genome. And that obviously has a chance to cause a ton of mutations. Luckily, those are very well repressed for the most part in our cells. Like our cells have, they methylate them and modify their histones to keep them closed off and keep them from being expressed. A lot of times they serve very important functions, and they've been kind of co-adapted or co-opted by our cells. So if you think about a chromosome, in the pictures, they're like these kind of X-looking things in our textbooks, and you'll find these repetitive elements, and a lot of those are transposable elements at like the center of the chromosome or on the very tips of the chromosomes, and so those serve very important functions. But these TEs, they can be broken up into two classes. So we don't have to go through all these mechanisms, but they depend in what's called their intermediate. So one is a DNA transposon, which I said makes up 3%. So that can basically move from one place or another without an RNA intermediate. and that's what separates line 1 elements. They're this class are an RNA class 1 retrotransposon, where they are expressed as RNA. They are made into a protein, which then encodes a reverse transcriptase, and they'll actually go in and reinsert themselves into the genome. But you have all these different classes. People have spent decades looking at this, and then those can be broken up into different families. So it's a pretty, robust and complicated area of research. But here we're thinking about specifically line 1 down here. So to kind of give you a better handle, I know this is just a lot of lines. I'll give you a better handle on it with this figure here, where, here's one copy of line 1, and there's, millions of copies of this. But it gets expressed like any other gene. So you have transcription factors that recognize it. You have polymerase that recognizes it. It expresses it into an RNA. That RNA, which is unique here, is it encodes for 2 different proteins. Typically, an RNA will only encode for 1 in mammalian cells, but line 1 encodes for 2. And they've been, RNA that are being translated are largely kind of nomenclated with open reading frame. So reading frames are regions that are being translated. So we have open reading frame 1 and open reading frame 2 protein. And those 2 proteins have very distinct functions. ORF1 will bind to the RNA, and ORF2 serves an enzymatic. So this helps protect the RNA. This line 1 RNA will then go back into the genome or the nucleus. They'll recognize a spot, and then that ORF2 protein will actually nick the DNA and then reverse transcribe that RNA back into DNA, and that's how you get that amplification of the gene. Again, this happens very rarely in our cells. This is not something that is, actively going on. And this process in general, this reverse transcriptase process, is very inefficient. You don't hardly ever fully reverse it. It gets stuck at some point, and you end up with a bunch of shortened copies throughout the genome. But in disease conditions, so this is where it's relevant, right? Why we care about this is in certain disease conditions, you don't see this upregulation of line 1 expression. Whether it's a cause or a symptom is a very open debate right now. More than likely, I'd say in the majority of cases, it's a symptom, meaning for whatever reason, this region of the genome is being relaxed, it's being made more accessible. And like we talked about earlier, it's a probabilistic type of thing. If it's more relaxed, then you have more access to these transcription factors that express it. But the expression itself, even if it is a symptom, can cause problems. So when you have this expression, you can have an upregulation of inflammatory pathways, of different enzymes that are active in the cell that can actually be kind of toxic to the cell and either cause cell death or cell malfunction, more or less. And so this is also seen in a number of cancers. It's also seen to happen as we age. And so as you get older, you start to have more expression of these. And this is typically given to the loss of that heterochromatin, of that repressed region. And so, I know I'm kind of rambling here, but in the sense of exercise, why we care about it is what we found out is when you exercise, you actually have a higher repression of these regions, and you end up with less expression of these genes. And so, we assume if there's less expression, there's less inflammatory pathways going on, and you have less of this process happening. And so if you think about an aging scenario, if you exercise, you have more heterochromatin, maybe that's keeping these things at bay, and maybe you have less aging symptoms. So we're constantly thinking about how we can increase that health span of ourselves and not just the lifespan.
Yeah, dude. So you comfortable saying that, and I know we're still early in this area of research, but I recall, even at Auburn, you were kind of connecting some dots with muscle aging, perhaps even sarcopenia. Line 1 activity seems to go up, yeah, and then exercise seems to. So this could be one of the mechanisms at play, right, with how exercise is a combatant against, the sarcopenic process, the aging process, and just muscle pathology is related to its suppression, its ability to suppress minimal activity.
Yeah. So, I think there was a paper that came out towards the end of our PhD that was kind of suggesting the same mechanism. So I mean, I would say if I had to have an opinion, I think that exercise would help to keep these down regulating aging, and it would help create a more healthy phenotype. This paper that I'm bringing up here came out in 2019, so it was just towards the end of mine. I think you already left by that point, where they knocked out the enzyme that helped repress line 1, right? So if you knock out the enzyme that keeps it repressed, it's going to be more highly expressed, right? So that's what this knockout, this KO, is referring to. Now, this is the amount of muscle mass here on the Y, and on the X, you have your different groups. So on the wild type, your hind leg muscle in this case was, around half a gram. Okay. When you knock out the enzyme that controls line 1, you significantly decrease that muscle mass. And when you give what are called reverse transcriptase inhibitors, so these actually block that enzyme from reverse transcribing that RNA to DNA. When you give those, you can somewhat rescue that muscle mass loss. You can at least ameliorate that loss that you're seeing in the knockouts, right? So so what does this mean? That SIRT6, the protein they knocked out, it does a lot of stuff. So it's hard to say specifically that it's only 9-1 is working on. But if you knock that out and then give these RT enzymes, you're talking about a specific process is involved with, and you're having this rescuing effect. So this is on mass, but also on fiber thickness as well, where they see, again, not a complete rescue, but at least an amelioration of the loss from the knockout animal when you give these reverse transcriptase inhibitors. So I think keeping this expression down is an important thing to consider, and I think does have symptoms that are related to muscle mass and health. And here's a couple of survival curves that they did. So in the wild type, obviously they last past 60 days, right? But if here's the survival percentage. So to explain this a little bit, the animals that survived, you can quantify a percentage of how many survived, how many died over the course of days. So wild type animals, overall healthy, all of them survive past 60 days, right? So you expect. When you knock out SIRT6, they're lasting 32, 33 days. Whenever you give these reverse transcriptase inhibitors, you're actually extending the lifespan of these knockout animals. So that doesn't speak to the overall lifespan. You're talking about 2-3 years of these mice. You're not saying that. What you are saying is that they die less early in the knockouts that have been treated with the reverse transcriptase inhibitors, and this is kind of used as a proxy. This may also be helpful in almost animals.
And so your title kind of gives it away here, but this seems to be, at least to me, pretty decent evidence that if we can decrease the activity on line 1, that seems to be a good thing for muscle, the retention of muscle, and it seems to be associated with a longer life, at least in mouse.
Yeah, I think, I want to be careful not to say like, oh, it's going to be, in this case, we'll stay with the example of mice. Like if you were to stop this from being expressed, that you would increase their lifespan, for sure. But what I think it at least does is it increases their health span, right? So let's say they still do live to 2 years. Well, maybe they're more functional at, 22 months versus with lower expression of this versus if they had it being highly expressed.
Yeah.
I do think it's involved. But just based on where the data is now, it's from to say, I want to be careful of saying like, yeah, for sure this is involved with shorter or longer lifespans. Also, because that's again not an entirely active area of research for the lab, so I got to be careful there. But I do think it's helpful to have these things decreased in expression. And here's a slide that kind of like hits on why it's important, where they have a lot of these different diseases that are associated with increased line 1 expression, right? So of course you have cell death, that's not good. But they also have cardiovascular disease, and have them down here. Alzheimer's disease is AD. You have age-related macular degeneration, cardiovascular disease, neurodegenerative disease, osteoarthritis, Parkinson's disease, rheumatoid arthritis. These are all associated with higher line 1 expression. And specifically higher line 1 expression in these inflammatory signaling pathways. So again, I don't think is necessarily the cause of aging. Really, I'm not saying that, right? But it seems to be a symptom of something that's going on. And so controlling these areas, putting them into more of a heterochromatic state, as a more repressed state, I think would overall be helpful to the cells. I think that's what I'm comfortable saying.
Yeah, that makes sense. Well, you mentioned sirtuin, and there's several. Can we make a connection here now, perhaps between NAD, which is a popular topic right now, either by ingestion of, and I don't know if you've seen any of Sinclair's work, David Sinclair. Have you looked at any of his stuff?
Yes. Yeah. Not a ton, but I've seen it. Yeah.
So, in simple terms, NAD levels decrease as we age. Possible that we could ingest NMN or NR, which and increase NAD levels and thereby increase the activity of the sirtuins. I'm just trying to make some connections here with what you just showed in line 1 activity. If Cert6, you said, if you knock it out, that increases line 1 activity and is associated with muscle loss. Is that right? Okay. And then you can supply these drugs that increase, you said, is it sirtuin activity or the activity of the RNA?
No.
Yeah.
Yeah. So what's really interesting about those is those are still in knockout animals. So you have no Cert6 in those animals.
Okay.
What you're doing is you're stopping the enzyme from line 1 working. So if you stop that enzymatic activity, able to rescue to a certain degree the muscle loss that you see with a complete knockout.
Yeah.
So it's knockout plus treatment.
Okay. So do you see any utility in either through intravenous infusion or the oral ingestion of something like NMN or NR as another form of this, as like NAD precursors? Do you think there's something to that?
I want there to be. I again, similar to when we were talking about DNA methylation, I am not as familiar with when you get an IV injection or if you ingest it orally. I'm not as familiar with what those concentrations look like in the tissue. So if those concentrations increase in the tissue, then presumably you'd have an effect on the enzymatic activity of, in this case, Cert6. Now, one thing that I'm not only the concentrations, but now we're thinking about how long those concentrations last, right?
Right.
So now you're talking about not only do you need to transfer that NMN or NR into the cell, now you have to have it around long enough for Cert6 to use it in the compartment it's necessary for.
Right.
So Cert6, I think, is active also in the cytoplasm. But now, here we're thinking about the nucleus. But now it's got to get to the nucleus and then allow Cert6 to use that as a cofactor to deacetylate histones. And then how long does that deacetylation last? So there's a lot of kind of intermediate steps that we have to think about that I'm not as familiar with. So those would be the things that I would, want to check on to think about, how useful these things would be. I've seen some studies where when they increase the concentration of these substrates, they do make a difference on histone deacetylation, but I can't remember the exact context of how they were looking at them and if they were, it matters if these were in cells or in vivo or if they were already prematurely, having some sort of disease condition or something, right? Like this all matters. And it's just an area that I'm not as familiar with, but there's a lot of intermediate steps that are important in this process.
Yeah. Well, and from some recent exploration of that area, I'm aware of no studies personally that have actually documented you could take perhaps like a gram of NMN and where tissue biopsies have been collected to see the effect in one tissue, that it's actually increasing in it.
Yeah.
We have a few studies showing elevations in the blood, but we're not sure the true bioavailability. And like you said, the importance here of getting that into the compartments of the cells where it needs to be available for the sirtuins to do their thing. We're not certain about yet. Okay, man, I could talk to you for hours. I really want to have you back if you would give us the honor and pleasure of doing so, because there's a lot of questions to be answered. I tell you what, in closing, I would just like to hear from you, man, in terms of exciting areas. I guess twofold to close it out. One, the research that you're currently doing, what are you most excited about right now? And then two, what are you looking forward to pursuing, in the near future? Where do you see your area of research really going?
Yeah, I think, one thing that we're really excited about is, and whatever we do, right, in our everyday lives, does that have some sort of effect on our chromatin? Okay, is that itself impacting our ability our bodies to remember things, to affect processes, more in a long-term state than a short-term state? We know these things happen short-term, right? Like we resistance train, we know mTOR signaling goes up, right? We know fat signaling goes up, but we don't know how long that lasts. And so for me, thinking about epigenetics, epigenetic traditionally defined as something that's a heritable change, right, to gene expression that is independent from the DNA sequence. So it's not a change in the sequence itself, but on top of it, epi is on top. So I would like to see how things that we do in everyday life can actually be maintained over time, possibly inherited. That's a pretty tough thing to prove in humans, but possibly inherited. And, I'm huge on sort of autonomy and our ability to affect things, right? I think sometimes I'm a little too confident in, my individual ability to affect things. So I'd rather view, people, myself or any other person as powerful entity than something that things just happen to, right? So I really want to understand what we can do to affect our health. So that's number one. And I think we're doing some of those studies now. We're looking at how chromatin is responding to things like exercise. We're looking at how chromatin is responding to things like inflammatory signals. And we're not the first lab to do that, obviously, but we are looking at it in a slightly different way by adding this three-dimensional component to it. The other side of that is, okay, so now we know how our daily lives or our daily activities affect our cells, our chromatin structure, our chromatin responses. Can we fine-tune those things, right? Can we actually make a change? And so that's another kind of exciting area where I hope things go in the future is, can we actually, come up with something that helps people who are bedridden and under a disease condition, having treatments, something that just enables them to exercise, right? Like exercise. I mean, we take for granted how easy it is to walk down the street, right? I worked in a lab where muscular dystrophy was a big focus, and like it's devastating to see what happens in somebody who has Duchenne muscular dystrophy, and we just take for granted that we can go into the gym tomorrow. And to a lesser degree, think about people who do suffer from metabolic diseases or cancer or some along those lines. Like everybody's kind of been touched by that, and you can see how much it impacts you. And so, I hope that we can try to create things, or at least uncover things that are going to be helpful in the treatment of these conditions. And I do think that exercise plays a unique role in this that is missed in other areas of traditional research. And so that's why I've pursued it the way that I have. So those are, I think, the two things most that we're excited about now and then in the future.
Very cool, man. Well, yeah, I definitely would like to have another interview here with you, man, and dig into some of the techniques, particularly like how very useful, slides and gave a lot of context, but how this stuff is actually studied, how sophisticated the lab methods are to create those sorts of visual representations, and how we actually measure these things. And part of Fidomics, the core really of how we try to help people is through testing. We want to test and not get say, and then leverage that information to intervene, particularly through exercise, lifestyle, nutrition. And so I love digging the lab methods, and we do a lot of blood testing, but the genetic testing and the commercially available assays versus how the stuff that you're doing, differentiating between those, I think would be really helpful content. Knowing how, for example, a 23andMe test works versus if we're looking at the 3D structure of DNA and epigenetics. These are different things. Well, buddy, how can we keep up with you, man? Do you have, I know you have obviously a Google Scholar and ResearchGate, but you're in the lab a lot. You're a professor, you're teaching courses, mentoring students, and you're a husband and a father. And so I understand you're not super active on social media. What's the best way for any listeners to connect with you, see your work? Where would you direct us?
So we're trying to put together our lab website now. So hopefully that'll be up in the next week or two. So essentially, if you just Google Romero Lab UCSC, you'll be able to find it. I do have a faculty page that has a link to all the publications as well. And same thing, you can just type up Matt Romero UCSC. But I'd say the website's going to be the best means. And on there, we'll have the techniques that we use, the papers we published, who's in the lab. So we're trying to make it kind of user-friendly, so that way, if you're interested in this sorts of things, you can actually ask, well, how do you look at this stuff? Okay, you say on your webpage, you do this, but how can we actually know what you're doing? So we're going to have some of the techniques on there. We're really trying to focus on science communication and effectively communicating what we're doing out there, so that way people aren't just thinking we're in some ivory tower making stuff up, but we're actually doing the nitty-gritty work, trying to uncover things.
I dig it, man. Are you accepting any graduate students?
I am, yeah. So right now, here it works a little differently than when we got our PhD. When we went, we just, talked to Dr. Roberts. We're like, hey, can we join? But they actually here, they have a rotating program. So you apply to the program, and then you pick which labs you want to rotate in for a short amount of time. And then you pick after that. So if anybody's, I'm always trying to recruit people. I have a couple people interested now. We'll see if they end up joining. But yeah, we're looking to recruit graduate students, postdocs as well to do some of this work. And it's I think it's going to be good. It's a lot of work. It's a lot of, if you come from the exercise world, it's a lot of lab techniques to wrap your head around. And if you're from the molecular biology world, there's a lot of physiology to wrap your head around. So we're trying to tie these two fields together.
Beautiful, man. Well, that's exciting. So yeah, any listeners that are exploring, graduate school, postdoc, that would be a really cool opportunity. Well, hey, man, thank you so much for your time. And we'll be talking to you again soon, man. We appreciate it.
Yeah, can't wait. Thanks, guys.
Yeah, appreciate it, Matt.